Good evening, everyone, and welcome to the second episode in the Fossil Project's Fossil Roadshow webinar series. Thank you so much for coming. I'm Sadie Mills, your moderator tonight. I joined the Fossil Project this fall as the new project coordinator. To give you a little bit of background, this webinar is part of a series where we'll be touring the country and making 4 stops along the way, showcasing 4 fossil clubs and their featured fossils. As always, our webinars are being offered as a partnership with the Paleontological Society and facilitated via iDigBio's Adobe Connect platform. There are 4 webinar episodes in this series. Follow the Fossil Project on Facebook and Twitter for details on the upcoming November and December webinars. You can watch a recording of the first episode in this series and learn more about the Fossil Project at www.myfossil.org. Now, before we get started, just a little bit of housekeeping. This webinar should end no later than 8 p.m. Eastern Time. No one except for the presenters and myself have an active microphone. If you can hear us and see the PowerPoint, you're good to go. Please know that you can type in the chat box at any time, but 2 to 3 questions will be addressed after we show each specimen, and then we'll address the rest once we've gone through all of the fossils. If you're having trouble with the technology, try exiting the program and re-entering Adobe Connect. Or you can type in the chat box for technical support and our team will help you out. Also, please don't forget to take our short survey after the webinar ends. It is really important for us for our reporting purposes to our funding agency, the National Science Foundation. So we truly appreciate your time in taking the survey. Additionally, this webinar is being recorded, and both the recording and the presentation will be made available under the resources section of our website, myfossil.org. Lastly, in case you don't already know, by attending all 4 webinars in this series, you can receive a certificate of completion. In order to earn the certificate, you must be signed in with your full name, remain and participate for the entire hour, and be a member of myfossil.org, our online community. And so with that, we thank you for attending tonight's webinar, and a big thank you to our speakers that are sharing their time, knowledge, and specimens with us. We'll start with a quick overview of how this webinar will be structured. We will begin with introductions of our featured expert and club members. Dr. Nestell will then go through a brief overview of Pennsylvanian fossils in North Texas. Next, we will start the fossil ID. Each fossil owner will give a little backstory on how they found their specimen. Then, Dr. Nestell will share more information about the fossil. There will be a short Q&A after each specimen. Please type any questions you have regarding the fossils in the chat box. After we take a few questions, we'll move on to the next fossil. After we've gone through all the fossils, we'll address more questions from the audience at the end. And now for some introductions. Our expert tonight is Dr. Merlynd Nestell, a professor of Earth and Environmental Science and Mathematics at the University of Texas at Arlington. He is also a member of the Academy of Distinguished Teachers and has published 45 research papers and abstracts within the last 5 years. Dr. Nestell's research interests include mid-continent to north-central Texas, late Pennsylvanian, and early Permian cyclical strata. We are very excited to have him with us tonight. We'd also like to welcome our featured club, the Dallas Paleontological Society. This club was founded in 1984 for the purpose of promoting interest in and knowledge of the science of paleontology. It was intended by the founding members that the society would be a network for the exchange of data between professionals and serious amateurs in this field. With us today, we have the club's president, Tom Dill, and member Bob Williams, both of whom have been instrumental in putting together this webinar. We also welcome DPS members Donna McCormick, Mark Sutton, and Laura Grimm, and thank them for sharing their time and fossils with us tonight. Let's get started with an overview of tonight's theme, Pennsylvanian fossils of North Texas. Dr. Nestell, I'm going to give you your microphone and you can begin. 1 moment.
Yeah.
All right, you can go ahead.
This first slide shows a map of this general area in north central Texas, and you can see the locality at Jacksboro right in the center of the slide. Most Pennsylvanian rocks are exposed in north central Texas that in this particular area go from south of mineral wells all the way up to into Clay County. If you go to the west, you're in Permian age rocks. If you go to the east, you're in Cretaceous age rocks. So the next slide. How do I get to the next one? This is the Jacksboro Spillway locality. This is Lost Creek Lake that you see right here. And then the major outcrop area is along this slope right here. It goes around like that. And this is a surface that would be at the bottom of this outcrop area. And you can see there are well gullied strata all the way along this side. You'll see in the next picture exactly what they look like. And then on the top, there is a limestone that's called the Jacksboro limestone. The way to get to this is to go across the spillway of the dam. It's in a park, so you have to pay a fee to go there, a very minor fee. But in any case, it's accessible with no problems. Next slide. This shows the outcrop. This is just part of it. It goes actually in a big arc, as you can see there. And you can collect anywhere along these slopes, and then of course out on the flat there's a lot of stuff that you can find there too. But it's well weathered. And then this is the Jacksboro limestone. It's very thin at this place because the top part of it's eroded away. It's about 25 feet thick, and in this area there are 3 major quarries that actually quarry this limestone. And we actually had a student here at UT Arlington that did a carbonate petrology thesis on this limestone. Next slide. This just shows how this outcrop looks. And so you can spend all your time searching along these gullies and find all sorts of goodies as far as the fossils are concerned. Next slide. This is a strat column from a student who actually did a thesis on the paleoecology of the finest shale at this locality. At the top we have the very basal part of the Jacksboro limestone, and at the bottom we have sandstone and a kind of a greenish colored clay. The important thing to note about this particular strat column is that we have exposed here a cycle. A cycle in the Pennsylvanian consists of basically strata that goes from non-marine to fairly deep water, maybe several hundred feet, to fairly shallow marine, and then the shallowing goes upwards, and then if you get above this about another 40 or 50 feet, you go back into sandstone again. We call this a cyclothem, and I'll show that in the next slide in a minute. But the point I want to make here is that we have basically a transgression of the ocean and then a regression of the ocean as you come back. This was done by glaciers forming in the made primarily in the southern hemisphere, and what happened was you had to make ice. First of all, if you make ice, does it take a long time to make ice or take a long time to get rid of ice? What do you think? Well, if you're going to get rid of the ice, you can melt it fairly fast. So the transgression is fairly thin, and then the regression is fairly thick because it takes a long time to build that ice back again. So the transgressive part of this cycle goes to right about there, and then the regressive part begins and continues on up until you get to sandstone at the top. One significant thing about this outcrop is that right at this locality right here, at this level I should say, there's a sequence of myelina banks. Myelina is a clam, and basically you had oyster banks just like we have modern oyster banks. And there's another one of these above the Jacksboro limestone, on the top of the Jacksboro limestone. And so these things are evidence of very shallow water. And then there's a very, very thin limey layer about 2 inches thick at this level right here, and then you go wham right into a black mudstone, which is fairly deep water. So there's probably been some erosion in this particular part of the section at this site. All right, the next slide. This is just a kind of a cartoon that shows a cycle. This is the deepening of the ocean, and this is the shallowing of the ocean. And then this just shows a distribution of all the different types of fossils. Conodonts, for example, offshore, near shore, and phase of deposition and so forth. Back down here you can see there's sandstone, limestone, and then this black shale is the deepest part of the cycle. And then we have now again a regression where quite a bit of material gets deposited back up to a sandstone again. And the two myelina banks at Jacksboro occur right here and right here. And that's evidence of this transgression regression. So this is part of the importance of this particular locality, is that there is very well exposed cyclothem, not at the finest shale spillway locality. You have to go around the corner on that lake to find the stuff above the Jacksboro limestone, but it's there not very far away, within about, maybe several hundred meters. You can find the rest of the section on private property. So that's about all I need to say about this. This fossil locality is a great locality to collect because when it rains, the mudstone breaks down, and so you find a lot of nice fossils there. We know a little bit more about this cycle because a professor at Texas Tech, James Barrick, and I are writing a paper with my wife on the microfossils, and we're concentrating on this little interval right here on the section.
Dr. Nestell, can I interrupt? Your cursor is stuck.
I didn't hear that.
The cursor is stuck.
Oh, the cursor is stuck.
Is it not moving?
Where's our cursor? Oh, there it is. Okay. We're working on the microfossils basically in this interval right here at the moment because it's very nice evidence of this transition, and there are a lot of nice conodonts, which are teeth of a non-existent organism today, and that's our expertise, and so we're writing a paper about that. That's why we're working on this particular area. But as I said, it's a very popular place to collect lots of wonderful fossils, which we're going to take a look at next.
Yep.
All right, thank you so much. Let's go ahead and move on to our main event, fossil identification. If you're in our audience, don't forget that you can ask questions in the chat box at any point, and we'll address a few after each fossil, but we'll try and address all of them as we go along. So, Dr. Nestell, do you want to tell us about this fossil?
Now, this is a rather interesting fossil that I found in the black shale at the base, in this black mudstone at the base, which we call a black shale. And these are Conyo Lairds that are found there. Now, Conyo Lairds are rather interesting organisms. Nobody really knows where they belong. They're put in with corals or cnidarians in the phylum Cnidaria, but this particular locality has lots of Conyo Lairds, and everybody who goes up there usually collects a lot of these Conyo Lairds. As I said, nobody really knows what these things are, but they are thought to be related to corals. They have four sides to them, and then you can see these nice growth lines. Well, I found this thing several years ago when my student was working up there, and so the first thing I thought was it's a Conyo Laird. It has the nice little grooves here that look like that, but notice it does not have the four sides. It's not a Conyo Laird. It happens to be a cephalopod. It's a nautiloid cephalopod, and it's Botarioceras. It's actually a genus that's fairly well known, but it looks like a Conyo Laird. So people would look at this and say, hey, I've got a big Conyo Laird, but not true. Any questions about that?
It looks like... Did we have a question in-house with you guys?
Yeah, why are they blue?
Oh, I don't know why they're blue. It's just the preservation, but they happen to be blue in nearly every locality in north central Texas. By the way, there are very few localities where you can find these Conyo Lairds. There's one down by Mineral Wells where if you go there you may find 2 or 3 Conyo Lairds. I do know a locality on private property south of Jacksboro where you can go and practically walk on them, but they're very, very scarce, except at this Jacksboro locality, and for some reason they're very abundant at this place. But in any case, they're blue for some reason that I don't know. It's a type of preservation. And by the way, these are small Conyo Lairds. They actually get big. I have one from Bolivia that's about 5 inches long, and so it's a big one. Any other questions?
Well, thank you so much for sharing. Since we don't have... It doesn't seem like we have any questions just yet, so we'll go ahead and move on to our next fossil. Next, joining us is DPS member Bob Williams. Welcome, Bob.
Thank you. I found this.
Can you tell us a little bit about this fossil?
No, but I'm hoping the doctor can. I found it out on the flats where he found Conyo Lairds, and just noticed a kind of cloverleaf shape to it. And I have seen a few up on the bank, but this one came from out in the flat area. And that's all I know.
That's all you know about this? I can tell you a little bit about this. First of all, the Conyo Lairds are generally found on the flat area because they are in that dark shale at the base of the section. That's where their normal locality is for them. And of course, that flat area has well exposed black shale interval. It's all over that flat area. So you would expect to find things that you would find in the black shale interval just above the flat area. And these things occur also in that dark interval. These are fairly deeper water corals. And there are a lot of these there. If you look around, you can find quite a few of them very easily. They are tabulate corals. Now, corals in the Paleozoic naturally fall into two piles: tabulate corals and rugose corals. Rugose corals are horn corals. We're going to look at some of those coming up. And some rugose corals are colonial, but very, very scarce. But tabulate corals are generally colonial, and this is a colonial coral. And for some reason, the environment was just right for Conyo Lairds and these things to live. And that's why you find a bunch of them there at this particular locale. But mainly on the flat, you'll find these because they were in that dark interval at the base of the section.
It didn't seem to have an attachment point like a lot of corals.
No, if you look on the underside of it, you'll see it comes down to kind of like a dish.
Yes.
It's like a saucer. And that's because it lived on a muddy substrate. It
wasn't attached.
And it wasn't attached, but it had a nice surface area on the bottom to keep it from sinking down in the mud.
Well, it looks like we have a question from Linda. She wants to know, how big is this fossil?
About the size of a penny or a little smaller. Maybe a dime. From right here to right here would be about the size of a dime.
Great, thank you.
Now, by the way, I should say that the coral polyps sat in this little part right here. Remember, corals are made of polyps, and so the soft part of the coral was right here. So this one had 4 little polyps sitting in it when it was alive.
Excellent. Thank you so much, Bob. Our next fossil. Oh, it sounds like we had another question on that fossil. They wanted to know, how did you find the fossil, Bob?
The flat area is overgrown partly, but there's some areas where it's just soil and very few rocks. So anything that's there really sticks out plainly. And that's, I just was walking on the way to the bank where most of the fossils are, and checking out everything I looked at, a lot of nodules, but once in a while you see something out there that's a fossil.
Excellent. We had another question. Can you tell us if this was solitary or a communal animal?
These were solitary. Well, there were 4 polyps in this particular little colony, but these things lived by themselves, and they evidently were just scattered around on the sea bottom. But no, not all joined together in a colony, like an oyster colony, for example.
They can be more than 4 or less than 4.
There are more than 4 or less than 4 of these things. I've seen some with 3, and I've seen some with 5, so it just depends.
Wonderful. Thank you again, Bob.
Thank you.
Most of them are fairly small.
Next, joining us is Laura. Welcome, Laura.
Hello.
Thank you for joining us. Can you tell us a little bit about these fossils?
Well, these are microfossils. I thought I had a slide with the scale, but obviously not. These were found in the black shale I dug approximately a foot below the grade level and took that shale home and soaked it and processed it and then picked through it under a microscope to find these particular fossils.
Excellent. Can you tell us a little bit more, Dr. Nestell?
Oh, yeah. We call these things hooks and wheels for obvious reasons, or flying saucers. But basically, these things look like fish hooks. Actually, the little hook is broken off right there. It's broken there. And then you have some things that look like sieves. There are these. And then we have
I
these
think your cursor is stuck again.
These things are the hard parts of holothurians. Now, holothurians are sea cucumbers, if anybody can know what those are. And people actually eat these sea cucumbers in different parts of the world. I've actually tasted a sea cucumber when I was in China one time. I didn't take a second bite.
So you're being brave.
It's like eating leather. But in any case, these things are about a few microns across. They're very, very small. We generally take pictures of these with a scanning electron microscope. And they are actually quite common in our local Pennsylvanian age rocks. They're also common in the geologic column all the way back to the Cambrian and up to the present, so they're not uncommon. They're an echinoderm, so they're kind of related to crinoids in a certain sense. But as I said, you can find these quite often, and there are actually a number of papers have been written. There was actually a student at Texas Tech who did a PhD dissertation on our local Pennsylvanian holothurian sclerites. But if you take just about any sample of our dark shale or mudstone and process it through breaking it down either with water or some other method, you'll find lots of these things generally in that residue.
I had a question looking at this. Most of the wheels are six-spoked, so to describe them, and then there's the one.
Now this one doesn't have. This has more than six.
Yeah, would that be a different species?
That would be a different species.
Okay.
But bear in mind that these
Can
things.
I interrupt for 1 second? We're not seeing the pointer on our end. Can you click the arrow and drag it so we can see which one has the different spokes? Perfect, thank you.
Different number of spokes right there. And this has 6 spokes. So these are related, these 3 or these 4 right here, and then this one's kind of a ringer in the crowd. But remember that these characters were the hard parts in the soft part of the holothurian.
Right.
They were in the body. And my wife actually worked on some of these with one of our biology instructors, and they went down to the local market and got a holothurian and cooked it and got rid of all the soft parts, and then took the sclerites, these are called sclerites, and actually took those and took a bunch of SEM pictures of these things. And on the journal Microscopy, they actually made the cover of that journal several years ago and illustrated some of these things.
So have the forms changed much?
Not a lot, but there are all kinds of these sclerites if you look at them. Just look up holothurian sclerites and you'll see there's a great variety of shapes and sizes of these things, but most of them very, very tiny. But they exist in present-day holothurians also.
Well, thank you. We do have a couple of questions for you. The first question is again about the spokes. So are the number of spokes how you tell apart different species?
They would be considered as different species, but remember, this is a case where you use the form species terminology. Remember, form species are things that an organism can have a number of the different things. A good example of that are conodonts, or your teeth. For many years, the taxonomy was such that each different type of a conodont was given a different generic name or a different species name. Now we're interested in making a construction of the apparatus of the conodont. It would be like taking all your teeth out, putting them down on a table, and then having somebody else come along and say, how would you put those back together? And in the meantime, naming them all different genera. So your incisors would be one genus, your bicuspids would be another genus, and so forth. Canines would be another genus. We use the term form species for these kinds of things. Trees have this problem, a lot of different parts of the tree. They all are given different names. So the Linnaean taxonomy kind of breaks down in these situations. But in any case, these would probably be all considered to be different, at least species or maybe different genera in the taxonomic situation.
Thank you. Our next question is, do we know what the sclerites were made of, what mineral?
Calcite. They're calcite.
Excellent. And then we had another question. Do we know what the holothurian looked like, apart from being related to the sea cucumber?
Yes, there's a very, very interesting YouTube video. It's called, if you want to take a look at it sometime, it's called the Sea Pig. And it'll greatly amuse you if you watch that video. So I'll just leave it at that. But they look like they're actually quite colored in the modern sea. Some of them are red, orange, green, and they just kind of look like a sack, an elongate sack.
And even now, there's lots and lots of different forms.
Lots of different forms
big
of
and
these
fat
things.
and thin and real skinny. And
Basically, they plow through the sediment, take mud in, take any nutrients out, and put it back out again. And but anyway, that video is an amusing video to watch. I mention it to my paleontology class each year.
Well, thank you very much for the video recommendation. And it looks like one participant, Matt Croxton, may have linked to it for us, so thank you very much. And thank you, Laura, for sharing your fossils.
you're welcome. Thank you.
Our next fossil is from another DPS member, Mark.
Hi, how are y'all?
Good, welcome. Go ahead and tell us about this fossil.
Well, I don't know much about it, but I found it up on the hillside there in Jacksboro, and I was lucky that I didn't have to clean this one up or anything. It was just like you see it.
So what do you want to know about this fossil?
Well, I want to know how big do they get, how they survive.
Oh, bryozoans this is the bryozoan, first of all, on a crinoid stem.
Mm-hmm.
And as you know, and most people who are watching this webinar know, crinoids, some people have called them plants incorrectly sometimes. They're really animals who, in many cases, were attached to the sea bottom. Some of them actually floated. But in any case, this thing that's called the crinoid stem was the device that kind of kept the thing attached. It's made up of a bunch of little discs, and those discs are found free. And some people actually make necklaces out of them. There are a couple of places in our local Pennsylvanian near Mineral Wells where you can actually find a lot of them with little holes in them. And I've actually had people go out there to collect them and make a necklace out of them. The bryozoans actually attached onto the outside of the crinoid stem. Bryozoans are many times attached, many times free specimens. We have a lot of different bryozoans in the fossil record. Some of them are called twiggy bryozoans, some of them are called lacy bryozoans. This doesn't fall into either one of those two categories, but bryozoan taxonomy is not a fun thing to deal with. But in any case, they did like to attach to things. In modern terminology, some people call them moss animals for that reason. That's a common name for them. But in any case, bryozoans are in the seas today, fairly common. They go back in the Paleozoic to the Ordovician. They're one of the only groups of major fossils that are not found in the Cambrian. Most other phyla appeared in the Cambrian, but bryozoans basically appeared in the Ordovician. And in our area, in the Pennsylvanian, our local Pennsylvanian rocks, you can find in many places these crinoid stems with bryozoans attached to them. You can also find what we call twiggy bryozoans that look like branching trees, and some of them actually get up to the size of your thumb. And at this Jacksboro site, around the corner near the airport, I know a locality where you can go and find them, and they're as big as your thumb, and maybe 4 or 5 inches long. And each one of the little holes here, let me get my arrow with my arrow here. Each one of the little holes that you see here had an animal living in it. And so this is a colony of a lot of little animals.
So did it do anything to hurt the...
Well, it didn't really do anything to hurt the crinoid stem because, or the crinoid had a top. At one end of this, there was the calyx of the crinoid, which was the business end of the crinoid, and then at the other end, it usually was attached to some place on the sea bottom. So this was just a case of a convenient place to live. These things are actually called epibionts in paleontological terminology.
Okay.
Well, thank you. We do have a couple of questions. The first question is there a good reference for identifying Pennsylvanian bryozoans?
Well, yes and no. R.C. Moore was a very famous paleontologist who wrote the book Moore, Lolliker, Fisher, Invertebrate Fossils. And if you really want to find out about Pennsylvanian fossils, I would look in that book, first of all, if you can get a hold of it. You can buy it regularly on Amazon or some other site for a reasonable price. We have about 15 of them in our paleontology laboratory that I've picked up for students over the years for anywhere from 10 to 30 bucks each. But it's a very nice book. R.C. Moore did a lot of work on Pennsylvanian bryozoans, and so I would look up R.C. Moore and Pennsylvanian bryozoans, and you'll find several publications by him, and then that book has a lot of stuff, a lot of information.
Thank you. Our next question is, do bryozoans provide any protection to the host?
Not really. They're just kind of parasites living on the outside of the crinoid.
We have another couple questions. Can you tell us what bryozoans eat?
Well, they were filter feeders. They actually had little tentacles attached to a thing called the lophophore, and they could extrude it out of their little place where they sat or pull it back in. So they were actually filter feeders, so they actually fed in the water column. So they didn't really like water that was too muddy. They liked to live in clean water. You don't find many bryozoans, for example, at Jacksboro, at this finest locality, in the lower part, in that black area. You find them generally up higher in the more transgressive part of the cycle.
Yeah, that's where I found about midways up.
Yeah, about midways up, you
yeah.
find them. And then the local limestone has quite a lot of bryozoans in it, because it was a fairly clean environment.
Mm-hmm.
Well, excellent. I think that's all the questions we have on this specimen for now. But if someone still has one, feel free to type it in. Thank you so much, Mark, for sharing.
Thank you. Appreciate it.
Our next fossil comes from Donna. Welcome, Donna.
Hello.
Go ahead and tell us a little bit about this fossil.
Well, me and my grandson were out hunting. It was in the Jacksboro Spillway. And to be honest, he is the one that found it, but I thought it looked so nice that I wanted to go ahead and show it.
You have any idea what it is?
A nautiloid?
It's a nautiloid, yeah. Notice that it's very smooth. If you look at it, it's a nice smooth shell. Ammonites and nautiloids are two of the major groups of mollusks that are important in the fossil record. Nautiloids persist to the present, and we have modern-day nautilus, which lives in the southwestern Pacific, only in the world. It's the only place in the world where it lives. But ammonites started in the Devonian and disappeared at the end of the Cretaceous with the dinosaurs. And ammonites have very fancy structures on the outside, which are the boundaries between the chambers. They're called suture patterns, and they're really torturous as they go across, especially as you get up into the Jurassic and Cretaceous. In the Paleozoic, they're fairly simple. But in any case, this has a smooth shell, and now you've got it oriented upside down. You should actually have it turned around so that this part is pointing out this way, and that's where the animal actually lived. And this animal actually had a head, and the living chamber is called the phragmacone, and after that living chamber, then there were some tentacles. And these things had a head, eyes, and tentacles, and a beak, so to speak, and they were actually carnivores. Now, you didn't say the size of this thing, but I'd guess it's about the size of a quarter. Is that correct?
Yeah, around 4 centimeters.
Yeah, it's about the size of a dime or a quarter. But that's very common size for these things in our local Pennsylvania. These were floaters.
Well,
they didn't live on the bottom.
So we do have a couple of questions about this fossil. or maybe about the area in general. We got a question from John that says, is the black shale exposed at the old spillway or exposed at the new one?
The black shale is well exposed at the basal part of the spillway. That's correct. And it's also exposed on the flat. A lot of it's exposed on the flat, especially if you're facing the slope, looking at the slope to the right, you go up section on the spillway surface. So you go up through the black shale into the stuff just above the black shale. If you go to the left, you'll go down into the black shale. That's what's so nice about collecting out on the flat. You can find a lot of different things on that flat area because you actually have a lot of surface area exposed, but a very, very thin layer vertically.
Now, you mentioned that this one would have been about the size of a dime or a quarter. Was that as large as they would get, or did some get bigger?
No, they get very large. We have some nautiloids in the Fort Worth area in the Cretaceous that are 6 to 8 inches across and fairly fat. And our present day nautilus that lives in the southwestern Pacific, they're generally about 6 to 8 to 10 inches across. The males are small, the females are bigger, and as I said, they're carnivores. They live in the water column and mainly around an island called Palau in the southwestern Pacific. That's where people go to actually find the recent ones. And then their shells wash up on beaches all the way to Madagascar, to the Aleutian Islands, to India, to Australia, and so forth. But their actual living area is today in the southwestern Pacific, only place in the world.
Thank you. We had another question, I believe, about the spillways. I didn't finish asking it. We had a comment from John who noted that some of the fauna in the old site is absent in the new site. Do you know anything about that?
Is he talking about the old site between the two lakes? If he's talking about that, the answer is yes. There is a difference because at the old site, which I have about 15 samples every foot taken when it was still exposed, you don't really get down to the black shale interval at that site. You're probably maybe 6 to 8 feet above the black shale interval at the old site. If he's talking about the site that's now underwater at the upper end of the lake.
Well, thank you. And thank you, Donna, and thank you to your grandson for sharing this fossil with us. Give him our regards, too.
Okay, thanks.
Our next fossil comes from DPS President Tom. Welcome, Tom.
Hello. How are you?
Doing great. Tell us about these fossils.
All right. Well, I picked something fairly abundant that I found a lot of, so that's why there's 4 or 5 specimens shown there. I found all these loose, as most of the fossils on the spillway are. They erode out of the shale. So I don't know exactly what level they tumble downhill a little bit. I think this is about a third of the way up. Is where I usually tend to find these things. I believe it's a Brachiopod. What else do I know about them? That's about it. So,
We can tell you all.
yeah.
kinds of
Okay.
things about this. Okay. Yes, it is a Brachiopod. First question I want to ask you is, what's the difference between a Brachiopod and a bivalve?
Am I to answer that? Okay.
Yeah.
Well, bivalves are pelecypods and clams kind of thing, and they're supposed to have.
What's the bottom line difference? We have somebody waving her hand over there.
The bivalves have equal shell sizes, where the
Lot of them.
brachiopods had two different, a top and a bottom, distinct top and bottom shells.
Also plane of symmetry.
Yeah.
Plane of symmetry in a Brachiopod is vertical through the beak. If you look right here, you can see that the plane of symmetry is like this.
Right.
Whereas in a bivalve, the plane of symmetry is generally along the hinge line.
Mm-hmm.
And then, of course, the guts are totally different for both of them. But what about this Brachiopod? This is Composita, and probably the species name is Subtilata. It's a very common Brachiopod in the Pennsylvanian, and actually that genus goes up into the Permian. But the most interesting thing about this Brachiopod is that it is a spiriferid brachiopod. And it doesn't look normally like a spiriferid brachiopod, but it has a lophophore structure inside that's spiriferid. If you could cut this thing down the middle and look at it, it has two kind of cone-shaped spiral structures that go out like this. Whoops, let me get my pointer over here to do it right. If you cut it down through the middle like this, and then there's a structure that goes like this, that's cone-shaped and kind of like concentric rings, and a structure that goes like this on the other side that has concentric rings. These are the supports for the lophophore, and they're called spiralia. And spiriferid brachiopods have that spiralia. So this actually belongs to that group, which is a little unusual. Now, I actually have some specimens in my laboratory which are silicified, which show that structure very clearly. And, but in any case, this is a very common Pennsylvanian brachiopod. You can see the growth lines as these lines that kind of come down like that. This brachiopod has a very prominent fold and sulcus. This is called the sulcus, and this is called the fold. It's sort of like an anticline and a syncline as far as the geology.
And I don't know if they included the photo of the opposite side, but it has a very nice little round hole.
Yeah, nice
in the beak, in the
pedicle
valve.
Yeah. These things live at the end. You don't see the hole, but these things lived on a little stalk that was attached to the bottom, and that stalk was called the pedicle, and the opening is called the pedicle opening. Mm-hmm. By the way, these specimens are all upside down. You have them upside down.
Well, I included another photo the other way. What kind of water conditions would they prefer? Would they?
Well, now these things, these things you will not find in that black shale.
Okay,
they didn't like that kind of an environment. That's correct. They wanted an environment that was more aerated and a little bit clearer.
Okay.
And so you find them upslope.
Yeah.
Upslope at this locality.
That's.
You can actually find not a lot of these, but quite commonly in the Jacksboro limestone as part of the limestone.
Another thing I think is interesting about these is they always come out so nice. They're always complete.
Oh, yeah.
Both valves.
Yeah.
It's my understanding that there's something about them that when they die, those valves tend to stay together.
Yeah, they stay together, and they were probably tight, and they actually weather out like nuts.
Yeah.
Did you have a question?
No, it's just the way that the muscles were inside
Yeah.
the animal when they died would contract and hold the shells together. Where the bivalve, when they die, they relax, and
That's right.
They often get separated.
Can you repeat that so the rest of us can hear?
Basically, it's the muscle structure. Brachiopods have muscles that hold the valve closed, and they have muscles that close the valve and muscles that open the valve. Whereas clams generally have muscles that hold the valve closed, and they depend on ligaments to open. I don't know whether you've noticed or not, but if you try to pry a limpet off of a rock on the ocean, you try to do it, he'll just squat down right on the rock. So what he does is tighten his muscle up. And of course, clams tighten their muscle up, and then gastropods come along and bore a hole in them and have lunch. That's one way they do it. And starfish feed on clams by actually putting their arms around the clam and start pulling, and they just keep pulling, and the clam eventually relaxes, and then the starfish has dinner.
Mm-hmm.
Well, we do have a couple of audience questions. The first is, Tom, can you remind us where you found these again?
These are particularly abundant along the slope. I don't find these in the flats, and that fits with Dr. Nestell's saying that we would not find these in the black shale at the bottom part of the outcrop. They're about, I think most of these I find about a third to a half the way up, but they do roll really well. They're kind of round, so I think every one probably has come down slope somewhat.
Yeah.
So I'm not sure exactly what level in that outcrop they're from.
Yeah, they're from about 2/3 of the way up is when they're most common.
Thank you. Our next question is, are the growth lines laid down yearly or seasonally?
That's a good question, and you ought to ask Doug Jones that at the University of Florida, because Doug actually looked at Mercenaria as a clam, and he looked at growth lines and was actually, I think he did a dissertation on that at the University of Florida. But there's certainly some evidence that these things were seasonal, and of course corals also have seasonal growth lines. Some people have argued that they might be related to tidal cycles and other things, or the moon cycles, but that's a very good question, and I'll refer that to the experts.
Well, we've had a volunteer to ask Doug tomorrow, so we may be able to get back to everyone with that. Our next question is about the order, and you might have to forgive my pronunciation, but the question is Atheridida no longer a valid order?
Atheris?
It's spelled...
Well, there's Orthida, and there's Spirifida.
I think it's just Atheridida.
Atheridida. In our paleontology, I don't really talk about the Atheridida. It may be a valid order, but I'm not a Brachiopod expert, so I'd have to look and find out. But I don't think that's an order in our textbook.
Well, thank you so much. I think that's all the questions we have on this fossil for now. Thank you so much, Tom.
You're welcome.
We do have just a few minutes left, so I'm going to skip ahead to a couple of bonus slides we prepared, some extra fossils. So we'll come back to our conclusion slide in a second. But Donna, we have another fossil of yours that if you wouldn't mind sharing, we'd love for people to learn about.
Yeah, let's talk about this fossil. This is a good one.
Sorry to put you on the spot.
I really don't know what that is, but I did find it in the same Jacksboro spillway, and it was very...
What do you think it is?
I don't know.
What phylum do you think it belongs to?
I'm not sure. Sorry.
Okay. It happens to be a Rugose coral. And by the way, the size of this is maybe 1.5 inches long, something like that, maybe 2 inches at the most. But the interesting thing about this particular fossil, if you look at it, is it has, first of all, a very nice shape here, and then it has a bunch of these little bumps all over it. See all these bumps? Mm-hmm. And then it has these striations, and then these are septa. And so this is a Rugose coral. Rugose coral actually has a columella in the center of it, right here. The polyp sat here in this part right here, the coral polyp. And this particular genus is called Lophophilidium spinosum. I wonder why it's called spinosum. And at Jacksboro, there are actually 2 Lophophilidiums that are fairly common. I had another one that I was going to take a picture of and maybe use it just to contrast this. It doesn't have any spines on it at all. And usually it's found down in the black shale. It's fairly dark. And my specimen is almost dark blue, whereas this one is more brown. This one was probably found up in the finest shale, maybe under the Jacksboro limestone, maybe not too far, whereas the other coral is found in deeper water, which means that these 2 corals lived in slightly different environments then. But this one liked the shallower water stuff, and this is a very, very nice example of a Rugose coral.
Well, thank you so much for sharing. We're nearing the end of our time, so now is a great time that if you have any questions about any of the specimens that we had, we can go ahead and answer those. We have a question that says that a person is going to northern New Mexico next month, and he hears that the outcrops there are Pennsylvanian. Would the fossils there be similar to what we've seen tonight?
I can tell you a lot about New Mexico. We did our field camp at Las Vegas, New Mexico for 20 years, so I know that area pretty well. If you're going to look for Pennsylvanian fossils, I would definitely look in the Las Vegas area. In particular, there's a hill called Holman Hill above a little town called Holman. And up along that hill, there are several places where you can find Not a lot of Pennsylvanian fossils, but they're there. And you can find corals and Rugose corals. I can tell you that as you climb the hill, about the 4th or 5th long road cut, is a very long road cut, and there's actually a driveway that goes down to a house to the left, and right at the entrance to where that driveway takes off to the right-hand side is a very nice place to collect Pennsylvanian fossils. You can also find plant remains in that area. If you're going to go up around the Taos area, east of Taos, there are Pennsylvanian age rocks there, but I don't know a lot about good fossil localities there. But in the Las Vegas area on the Mineral Hill Road, there's good Pennsylvanian rocks there, and also along the Mora River, just to the east of Mora, there's quite a long section of Pennsylvanian age rocks there too. So those are places to take a look.
Well, I think we had a question about the first image that you showed that had the Conyo Laird and the cephalopod. I think maybe the pointer wasn't working and it wasn't clear which one was which.
Let's go back.
Yeah, I'll go ahead and take us back.
Okay, these are the Conyo Lairds right here. And this is the fake Conyo Laird, so to speak. It's not a Conyo Laird. It's actually a straight cephalopod. And straight cephalopods in the Pennsylvanian quite frequently are smaller than this, and they just look like tubes, tapered tubes. And they're called orthoceras. That's one of the generic names of them. And they're fairly common in our local Pennsylvania. These things are not common. I only know of a few specimens. This is the only specimen I've found at Jacksboro, this one right here. But one of our participants tonight actually had another one that's brown. That probably came from a little bit higher than where this one came from. Preservation is more brown.
Well, thank you very much. I'm going to go ahead and scroll to our conclusion slide because we're nearing the end of our presentation time. So bear with me while I move these. Well, I want to extend a very heartfelt thank you to each of our presenters and to Dr. Nestell. We want to thank you for your time, your beautiful specimens, and your knowledge. I know we've all really enjoyed them. Before everyone leaves, just a few quick notes. Don't forget to follow us on Facebook and Twitter for further information on our next webinar. We will be taking the recording that we've made of this episode and we'll post it to myfossil.org so you can come back and look at the fossils again. Please also don't forget to take our short survey and let us know what you thought of this episode. We really appreciate your time and the feedback that you give us. You can access the survey by clicking on the link. And it's also in the link in the chat box as well. Well, once again, thank you to everyone for participating. We're so glad to have had you, and we hope to see you next month.